Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Risk Assessment of Novel Neurotherapeutics, Mechanistic Translation of Preclinical Safety and PK/PD data to a Clinical Setting. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and they work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Allucent, who developed the content for this presentation. Allucent is on a mission to help bring new therapies to light by solving the distinct challenges of small and mid-sized biotech companies. The company is purpose-built through the convergence of leading providers to address this unmet need. Today, Allucent is a global provider of comprehensive drug development solutions, including consulting, clinical operations, biometrics, and clinical pharmacology across a variety of therapeutic areas. Now, I would like to introduce our speakers for today's event. Dr. Delat served as a senior pharmacology toxicology reviewer at the US FDA for 12 years. He is an established expert in translating non-clinical data to inform clinical program design, including dose selection, safety monitoring, inclusion-exclusion criteria for clinical protocols, and informed consent forms. Dr. Delat has an extensive experience in creating and implementing strategies for regulatory pathways in the US. Dr. Welty has over 30 years of extensive experience in preclinical and clinical pharmacology, leading organizations and project teams from early discovery to approval and commercialization in multiple therapeutic areas and modalities. Dr. Welty's primary therapeutic area of expertise includes neurology, ophthalmology, and rare diseases. He has extensive experience in pharmacokinetics, pharmacodynamics, and system pharmacology, and adopting model-informed approaches for candidate nomination and clinical development. And Dr. Wakeford has over a decade of experience in cannabinoid, psychostimulant, and entactogenic pharmacology, including 13 manuscripts on non-clinical drug abuse liability across all three classes of drugs. Her non-clinical expertise includes both rodent and non-human primate experimental design and analysis of these compounds with a specific focus on central nervous system effects. But now, without further ado, I'd like to hand the mic over to our first speaker for today, Dr. Alison Wakeford. You may begin when you're ready.
As Ryan mentioned, my name is Alison Wakeford, and I am going to be walking through an introduction of the product landscape related to CNS therapeutics. And before I get started, I wanted to go ahead and give just a quick thank you to Rachel and Angela, who really helped provide some of this incredible content that I think is going to really set up this product development landscape, regulatory changes associated with it, and challenges that we have so that we can really set up for our speakers, Dr. Delat and Dr. Welty. So the three things that I want to focus on today with regards to CNS product development is the current and the future CNS therapeutic market data that we have, as well as some key regulatory developments that really highlight the necessity of strong non-clinical assessments to be able to develop both safe and effective CNS therapeutics. And I'm going to round out this discussion with talking about the actual challenges that we have in the product development as well, to sort of give ourselves a better idea of where we are and where we can go forward. So as you guys can see, from around 2010, we see a really nice steady increase in the historical number of mergers and acquisitions within the global neuroscience space. And when we sort of zoom out of this solely mergers and acquisitions information, what we see, if you look at the second infographic, is that in 2022, the global neuroscience market split was around $612 billion, with about $159.5 billion dedicated specifically to drug therapies for both neurological and neuropsychiatric illnesses. And this growth is only projected to be astronomical in 2026, where we have an estimated $194.4 billion just for looking at drug therapies related to both neurological and neuropsychiatric illnesses. So looking solely at market data, we can see that we are really well set up for developing new CNS therapeutics for a whole host of patient populations. However, with that said, there are some key regulatory developments that are necessary to go over to sort of understand why non-clinical assessments are going to be so key in getting us to that projected growth. First and foremost, it's really important to state upfront that developing and regulating these neurotherapeutics is a complicated process that is due to both safety and regulatory challenges. A really great example of that is that in the past we have had controversial product approvals. These include products that over time have shown modest efficacy and severe toxicities. And this is specific to not only neuropsychiatric illnesses, but a really prominent example is with Alzheimer's and the occurrence of certain edemas related to therapeutics that are specific to Alzheimer's. Complicating this process is the non-human primate shortage that has affected the world. This has largely been as a result of the COVID global pandemic, as this pandemic sort of funneled and necessitated the use for non-human primates specifically for the development of COVID-related vaccines. But it also highlighted globally that an increased need for alternative models in non-clinical safety assessments. Around 2017, we also saw that there were several FDA label updates that were related to certain anesthetics, mostly of which were inhaled, such as nitrous oxide and isoflurane, that showed age-related neurotoxicities, something that the FDA obviously has a very close interest in and is something that is currently ongoing as far as whether or not those changes are taking place now or later. And finally, the opioid crisis, which again is a global concern, which has really focused and challenged regulatory bodies to really highlight the need for abuse liability non-clinical assessments, as well as developing abuse deterrent formulations. Now, these are obviously not all of the key regulatory developments that are, part of this process. These are just four major components that we've talked about. And in addition to these regulatory components, what we need to talk about is the actual challenges to developing these therapeutics. First and foremost is that the major challenge here is penetrating the CNS. And that's mostly due to the blood-brain barrier, which acts as a functional and physical barrier to protect the brain parenchyma as well as CSF. Now, in order for neurotherapeutics to be therapeutic, they actually have to cross this physical barrier. And what we know from radiation exposure, diseases, and other drug interactions is that the integrity of this physical barrier can be manipulated. And so in doing that, that has provided us with insight on new drug design, so developing drug molecules that are very small or that are lipophilic, as well as those that are nonpolar, so that they have an easier time crossing this blood-brain barrier. This has also led to us utilizing different drug delivery systems, such as intrathecal, intraarterial, in order to get these drugs to pass through this very tightly and well-controlled physical barrier. On top of being able to penetrate CNS, another major challenge, which is honestly a new opportunity, it's not a new opportunity, but an opportunity for the development of therapeutics, is that there is a huge global unmet need for these drugs and for these therapies. There is a global increase in incidence of mental health and disorders, as well as in neurodegenerative disorders. And recently, the World Health Organization showed that 1 in 4 people worldwide suffer from these disorders, and that potentially a growing increase in the incidence of these neurodevelopmental and neurodegenerative disorders is largely due to an increase in the human lifespan. And In tandem with that is the actual physical development of these. We need to be able to develop both safe and efficacious drugs. Because these drugs work specifically on the central nervous system, we have to be able to use correct non-clinical assessments in order to be able to assess neurotoxicities as well as seizure risks. Seizures are a very obviously important, but also a life-threatening challenge. And these are obviously things that we have to keep in mind when we are developing safe therapeutics. Unfortunately, part of the global landscape with regards to efficacy in central nervous system therapeutics is that there is a high fail rate historically. That mostly has been due to changes to limited permeability in the blood-brain barrier, which we just discussed, as well as the complexity of etiology of human diseases and the nervous system. So coupled together with the regulatory challenges that we talked about, as well as the changing landscape in the market for global neuroscience products, I'm really excited, and I hope that we have presented an overall introduction to help give ourselves a better understanding of where we are and where we can go. And I'm really excited to hand this over to Dr. Delat, and am very excited to see what Dr. Delat has to offer for us today.
Thank you, Allison. I appreciate the nice intro, and also want to thank everyone for showing up today. So if we can advance the slides. There we go. So the title of my talk today is CNS Risk Assessment, Identifying and Managing Toxicities Using Traditional Approaches. Next slide, please. So this lecture is going to involve a discussion of types of non-clinical data used in the evaluation of drugs, CNS risk assessment framework, CNS risk management framework, and then end with an overall discussion of conclusions. Next slide, please. So non-clinical data are used in various types of non-clinical data are used in the evaluation of drugs. This includes pharmacology, safety pharmacology, ADME, as well as toxicology data. Next slide, please. All of these data are very, very important in informing the risk assessment framework that we're going to talk about in the next couple of slides. Next slide, please. Now, before we jump into the framework, there are a couple of key definitions. Number one, hazards. Intrinsic toxic properties of an agent. Next slide. Risk. Probability of a toxicity based on an agent's exposure and potency. Next slide, please. And then finally, risk assessment, which is the evaluation of risk based on various factors that include hazard characterization, dose response evaluation, and agent exposure level. Now, it's very important whenever we're doing a tox study that we use multiple dose levels to characterize a particular hazard, right? So just remember that that's going to be a theme that's going to come up multiple times throughout the talk. Next slide, please. When we talk about risk assessment, there are many, many different objectives. In particular, we want to protect human health, establish risk based on toxic exposures, and we want to balance risk and benefits. Risk and benefit balance is very important, and it's a concept that's going to come up multiple times in this particular talk, and it's something that all sponsors should focus heavily on, especially when they know they have a compound that has toxicities. Next slide, please. When we're doing risk assessments, there are a number of key steps. Number one, start off by identifying the risk. We're going to establish if the risk is drug-related. And then next, we want to verify if this risk is biologically plausible. And we can do this in a number of ways. Number one, does the compound cross the blood-brain barrier in order to produce this neurotoxicity that we're seeing? Number two, does it interact with targets known to induce the risk of concern? And then lastly, does it belong to a class that produces the risk of concern? And then the last step is determining if the risk is clinically relevant. Now, based on my experience as a consultant as well as a former FDA reviewer, these last two steps are very important steps that a lot of people actually overlook, and it can actually have very negative impacts on your program. So pay very close attention to whether or not these effects are biologically plausible and if they're clinically relevant. Next slide, please. In terms of risk characterization, there's a lot of different types of information that's usually needed. Mechanism, mode of action, target organ of toxicity, clinical relevance of toxicity, exposure data, and most importantly, safety margins, right? So if you have a toxic compound, you want to make sure that there's safety margins that exist. Next slide, please. Now I want to move into talking about non-clinical findings that are actually indicative of CNS risk. Next slide, please. So if we think about product target sites, we know that there are a number of different sites that actually modulate, mediate neurotoxic effects. This includes sodium channels, adenosine receptors, CB1 receptors. Next slide. In terms of behavioral observations that are indicative of CNS risk, this includes seizures, convulsions, impairments in learning and memory. And as a graduate student, I did a lot of research to understand the disruptive effects of different drugs on learning and memory. So I can tell you behavioral observations are very important in understanding CNS risk. Next slide, please. Then we go to physiological findings, such as changes in electrical activity in the brain. Next slide. And then, of course, pathological findings that are indicative of CNS risk. And this includes neuronal damage, death, and adverse histopathology. Next slide, please. So the bottom line here is this particular slide shows us that there are a variety of non-clinical findings that are indicative of CNS risk. Next slide, please. No matter what toxicity you have that's drug-induced, we always want to create a profile for that toxicity. In this particular case, there are a number of factors that I want to point out when you're considering developing a profile for neurotoxicities. Number one, toxicity timing. This involves things such as onset of effect, offset of effect. Then there's toxicity pattern. So if I'm looking at an EEG record and I see stuff like a spike or a sharp wave, that serves as evidence that a seizure is probably occurring. And then there are premonitory prodromal signs, whether or not they're exhibited. So these signs can be exhibited in a dose-related manner, and they're exhibited prior to seizures or convulsions, okay? And then lastly, an important factor is exposure level at which toxicity occurs. So we definitely want to look at exposure level or know the exposure level at the no observed adverse effect level, which is used to set safety margins. But we also want to understand the shape of those exposure effect curves. So this goes back to the point we need to test at least 3 doses or more so that we can create a curve. And so we want to understand how that curve looks, whether it's shallow or steep, because the curve shape actually impacts how conservative we may be when we're managing risk. So for example, a very steep curve may call for us to take more conservative approaches to managing risk compared to a shallow exposure effect curve. Next slide, please. Now, this is really a very important part of the talk, which focuses on challenges related to assessing CNS risk. So all risks are not the same, right? Some are more challenging than others to assess, but we want to highlight different factors, different things that could make it more, in general, more difficult to assess CNS risk. Number one, there are numerous factors that can impact an individual's susceptibility to neurotoxicity. For example, physiological changes. This could include an animal being deficient of certain electrolytes, which allows it to be more susceptible to drug-induced seizures. Other factors, dose schedule, environmental stressors if they're present, and of course, age is a very important factor. Imagine an animal, an organism that's very young that doesn't have an intact blood-brain barrier, where the levels of the drug that enters the brain are much higher probably than that of an adult organism with an intact blood-brain barrier. So we need to look very closely at factors such as age. Other challenges, the profile may change after multiple treatments. So tolerance can develop, kindled convulsions can occur. And so kindled convulsions, for example, occur when subconvulsive doses are repeatedly administered over time to an organism. And then, of course, things such as seizures, convulsions, CNS lesions, they may actually occur in the absence of treatment, right? So it's very important for us to have a very good handle, very good understanding of our historical control data to understand what types of findings may actually be background findings compared to what may actually be drug related, right? So the reality here is there are a number of challenges that can impact assessing risk. Please keep them in mind when you're looking at your data and you're trying to understand what risk exists with your product. Next slide, please. So now that we understand these different challenges, the factors that contribute to creating a profile for your toxicities, let's jump into case studies. So case number 1 has been modified for blinding from experiences with new molecular entities with limited or no clinical history. Our primary challenge is, what is the NOEL, the no observed adverse effect level in the study that's going to be described. And then there's some key background information. So the species tested is monkey. Route of administration is intravenous infusion. There are 3 doses that are actually studied, which is important. And what we've seen data-wise is there are tremors observed in multiple animals, multiple monkeys, and one animal actually exhibited a convulsion during a pharmacokinetic study. Next slide, please. So in this example, we have a pharmacokinetic study where we actually are seeing toxicity. So again, data from different types of studies can be used to evaluate drugs, right? So the table that's here is a very busy table, so I'm going to walk you all through it. The first column is clinical signs. The next 3 columns are the number of monkeys that exhibited clinical signs. So we can see with the red boxes, there are 2 drug-related effects that occurred. Tremors occurred at the medium and high dose, and then a convulsion occurred at the high dose. Now, because these tremors occurred with a compound that produced convulsions, they're going to be viewed as premonitory signs of convulsions. So what I need everyone in the crowd to think about is, are these tremors actually considered toxic or not, right? Next slide. So based on my experience as an FDA reviewer and certainly serving as a consultant, what I've learned is that tremors are actually considered toxic if they're premonitory signs of convulsions. They're considered toxic unless proven otherwise. And one way of proving otherwise is if you conduct an EEG study to show that those premonitory signs, if they do occur, they're not occurring with abnormal EEG patterns, right? So there needs to be evidence to support you concluding that those premonitory signs are not toxic. But in this case here, with the limited data that we have from the PK study, the tremors are going to be considered toxic along with the convulsions. So given that we see toxicities at the mid and high dose, the NOEL will be estimated at the low dose, which is 3 milligrams per kilogram. I have to tell you, an important lesson here is, do not dismiss premonitory signs of convulsions. A lot of people have dismissed them in my career, and it's led to them going on clinical hold because they realize after talking to the agency, or the agency informs them that, hey, you have these premonitory signs, they're actually toxic, and in some instances you don't have a NOEL. So pay attention to premonitory signs of convulsions. They are toxic unless proven otherwise through EEG studies. Next slide, please. So things to remember with CNS risk assessment. They typically require various types of data to complete an assessment. Next slide. We want to use basic principles of science to make judgments. So for example, we determine if the onset offset of the toxicity correlate with pharmacokinetic, toxicokinetic profiles for the parent drug and a metabolite. An important question to ask yourself is, does the onset of the convulsions overlap with the Tmax, time to the maximum peak plasma level of the parent drug or its metabolites? Answering this question is important because it may allow you to understand what molecule is actually driving the convulsion. And then lastly, the final thing to remember is CNS toxicities can be exhibited as adverse alterations in a variety of things that include behaviors, physiology, and histopathology. Next slide, please. So the next step, we want to talk about CNS risk management framework. Next slide. There are multiple ways of managing risks. Setting safety margins, limiting patient populations, and implementing safety monitoring. Next slide, please. So in order to set an adequate safety margin, the margin should be based on exposure levels in the most sensitive species. An important rule of thumb, the most sensitive species is always the most relevant species unless shown otherwise, demonstrated to be otherwise. All right, never forget that. Next slide, please. So in case number 2, the question that we have is which species is the most sensitive? In the table that we have here, first column shows that we have 2 species, rabbit, rat. We see the target organs, the brain, the toxicity of seizure, and then we have exposure levels at the Noel. Based on these data, we can see that the exposure levels at the Noel are lower in the rabbit. Therefore, the rabbit's the most sensitive species. Next slide. Given this, the rabbit's data are going to be used to set the safety margins for clinical exposure levels. Now, important lesson here, whenever we have severe toxicities, or especially if we don't know the mechanism by which they occur, we typically want to have a 10-fold or higher safety margin. Another thing that we will do to protect patients is we'll establish top PK exposure levels. So the top Cmax in this case will be 1 nanogram per mil, and then the AUC will be 123. Next slide, please. We can also limit patient population by counter-interacting the product in patients that present an increased risk to expected drug-induced toxicities. The patients that administer the drugs with a toxicity profile that overlaps with the therapeutic product, as well as patients that present a form of disease in which alternative treatments are available that offer a better risk-benefit profile. So this last point is very important. We always want to do a risk-benefit analysis. If you have a drug that produces a toxicity, we want to show the agency that the benefits outweigh the risk. This is very important. Next slide, please. The other step we can take is safety monitoring. So if we have a drug that produces neurotoxicity, monitoring that could be used could be an EEG, MRI, self-report data. Next slide, please. So things to remember for CNS risk management: rely on findings that are clinically relevant and obtained from the most sensitive species. Next slide. We also have multiple ways of managing those risks, which is shown here: providing safety monitoring, limiting patient population, and setting adequate safety margins. Next slide, please. Now, this all brings us to our overall conclusions, which there are 2. Number 1, non-clinical data define the risk profile of therapeutic products that are defining the risk profile for therapeutic products are critical for informing the design of clinical studies and managing CNS hazards and other toxicities. Bottom line here, non-clinical data are important, especially if you're dealing with a new molecular entity. Point number 2, scenarios below illustrate how non-clinical data may be leveraged to inform the clinical design, the design of clinical protocols. So we're looking at dose selection. The dose should not exceed, preferably the exposure at the Noel, or if needed, the Noel when expressed as body surface area. And of course, this is in the most sensitive species, as always. If we're looking at inclusion-exclusion criteria, and let's say you have a toxicity such as seizures, we can exclude volunteers that have a history of head injury or seizures, or that are taking medications that decrease seizure threshold. And then finally, in terms of safety monitoring, we can implement EEG, MRI, or safety report data. So at this moment, I want to stop to thank you all again for giving us your time and your attention. And I want to shift over the mic to my colleague, Dr. Devin Welty, who will continue our webinar. Thanks again, everyone.
Hi, and thank you very much, Marcus, and to Allucent for such a great introduction to this webinar. In the remaining time, I will present a real case study that we worked on with the sponsor. Next slide. And this case study will go through a description of the drug candidate and some of the preclinical findings, and then talk a little bit about some of the model-informed ways that we used to make decisions on its continued development and in developing a first-in-human protocol design and some of the considerations in that clinical study. So next slide, please. This was a small molecule inhibitor of a novel target, receptor interacting kinase 1, or RIPK1, and the indication was for progressive multiple sclerosis. The RIPK1 pathway has effects on both the neurodegenerative and the neuroinflammatory pathways that are hallmarks of MS, and therefore was a very attractive, albeit an unvalidated clinical target to approach with this molecule. Importantly, it was believed that for pharmacodynamic effects and for efficacy, that the enzyme would need to be inhibited greater than 90% in brain over a steady state dosing interval. So we were really trying to put the brakes on this enzyme as a way to provide a neurotherapeutic effect. Next slide. With respect to the pharmacokinetics, this molecule, this drug candidate, had predicted very favorable human PK. And that is, it was expected to have very rapid oral absorption with bioavailability greater than 70%. The kinetics were linear, dose proportional, and the half-life was estimated to be around 3 to 5 hours. And this was important because it gave us the opportunity to potentially dose this once a day. And for a chronic med, this was very important. The drug was cleared primarily by metabolism and by multiple pathways in the CYP450 family. And therefore, its potential for being a victim of drug-drug interactions was low. Further, it had low plasma protein binding, offering a great population of unbound drug to get into the brain. However, there were conflicting data on brain penetration, and that is in some cell systems, the candidate was a PGP efflux substrate. And so it was very important for us as we began to think about how we would develop this drug to ensure that there was good brain penetration and to demonstrate that in the clinic to de-risk the program. As Allison and Marcus emphasized, the importance of ensuring brain penetration for a neurotherapeutic. Next slide. In safety assessment, the key finding that we ran up against was evidence of seizures in the tox studies. And this occurred in high dose rat and monkey dose range finding studies. This finding appeared to be associated with the Cmax. And in rat, in monkey GLP 28-day studies, there was absolutely no evidence of seizure findings. Nevertheless, in discussions with the FDA, it was decided that there would be an additional 10x safety factor associated with the NOAL Cmax of the most sensitive species, the primate, and this limited then our threshold for clinical exposure to 2,000 nanograms per mil. The key question for us then was it possible to achieve the clinical pharmacodynamic objective within this Cmax limit? And to answer this question, we needed to turn to a way of predicting brain concentrations and effect. Next slide, please. To do this, we used modeling and simulation for a clinical dose projection. We built a physiologically based pharmacokinetic model. Building from the ground up, we predicted first the plasma concentrations of each animal in the program. When we were comfortable that we could predict the plasma PK, then we added in compartments for the brain interstitial fluid, and cerebral spinal fluid. And once we then predicted the brain intracellular concentrations. And this is important because this is where the enzyme target would be and where we would need to deliver the drug to inhibit the RIPK1 enzyme. Next slide. And using this model, then we were able to simulate in a healthy volunteer population the clinical brain RIPK1 inhibition. I'm looking at these panels going from left to right at a 20 milligram dose once a day. We were not able to achieve the target, which is the red line going through the graphs at 90% RIPK1 inhibition. At 60 milligrams, we were able to just reach the target, but by 24 hours, many of the patients would not have coverage and inhibition of the enzyme. But by 180 milligrams once a day, we believe that we would be covering the entire population and inhibiting the enzyme 24 hours a day, greater than 90% at steady state over a dosing interval. Importantly, the plasma Cmax that we were able to achieve these brain inhibitions from this predicted scenario were within the 2,000 nanogram per mil Cmax limit. So this gave us confidence that we could go forward in development, and using these doses would help us to define the clinical protocol. So next slide, please. And this was our clinical protocol design and some of the considerations. As noted in our first cohort, we used a 20 milligram starting dose. We then escalated to a 60 milligram dose. And we left the third cohort dose as to be determined because we wanted to see the data before we escalated any further. In order to incorporate safeguards for the patients due to the seizure potential, we added a couple of features. First, we had a sentinel dosing. So one patient was given an active in each cohort to begin the cohort dosing. The sentinel patient, once it was determined was safe, and tolerated the drug, then we instituted staggered dosing in the rest of the patients in the cohort. Secondly, for dose escalation decisions by the Safety Review Committee, we looked at both safety as well as the PK and exposure and the next dose predicted Cmax. We also implemented a stopping criteria at Cmax 2,000 nanograms per ml. Secondly, we had another cohort, Part B. And this cohort was designed to verify CNS penetration. This was an open label cohort for CSF sampling and was at the highest dose. The highest dose was 120 milligrams. In this cohort, the subjects experienced 2 lumbar punctures by an experienced neurologist, and we chose 12 hours apart, 1 and 13 hours for 3 subjects, and 4 and 16 hours for an additional 3 subjects post-dose. And the reason why we use this dosing and sampling paradigm was to get some sense of a pharmacokinetic profile within the CSF as relationship to plasma. Next slide, please. And here was the outcome of this phase 1 SADS safety and PK study. Overall, the investigational drug was very well tolerated in the healthy volunteer population. There were no severe adverse effects, and there were actually very few adverse effects. It was one of the cleanest drugs we had worked on. The PK was dose proportional and safely reached the Cmax limit at 120 milligrams. The lower dose that we reached in this study was due to what we believed to be even greater bioavailability than 70%. And so we were able to achieve the Cmax limit at 120 milligrams. At this dose, the drug readily penetrated the CSF, and the drug was equivalent in the CSF to the unbound fraction in blood. And as we can see in the graph on the right, where the subjects' samples were scattered, that the drug entered the CSF rapidly and then began to decline in parallel to the plasma concentrations. Further, we were able to verify the pharmacodynamic target in blood and verified this, that it was inhibiting the enzyme as proof of concept. Having both an understanding of relating the plasma concentration to the brain and knowing how much was inhibited in blood, we could then project that the drug was indeed inhibiting RIPK1 in the brain at the target level that we were aiming for. We were also able to take the model and optimize it to inform future decisions in the clinical program. Next slide. Overall conclusions from this case study and for neurotherapeutics in general is that implementing an integrated non-clinical development plan is needed to effectively support clinical development. Further, model-informed approaches are valuable in incorporating the diverse set of data that come with a preclinical program for translation into clinical decision-making and study designs. And then finally, for first-in-human studies, these can be designed to de-risk key safety and CNS PK/PD aspects for further clinical development. Next slide. So I would just like to thank you and thank the audience for your participation in this. And if you have any questions, please feel free to bring them forward. We would love to hear from you.
Yes, thank you very much, everyone, for that very insightful presentation. I'd like to invite our audience to continue to send their questions or comments right now using the questions window for this Q&A portion of our webinar. Now, I've already received some questions from the audience, so we'll get ourselves started with those. The very first question that I have here, and it's for our first section, I think, Allison, you might want to comment on this. The question is, what are some major brain disorders that account for the current CNS market value?
Hi, Ryan. Thank you so much for that question. So neurological disorders such as multiple sclerosis, as well as common neuropsychiatric disorders such as anxiety, mood disorders, and substance abuse disorders currently account for approximately 61% of the market revenue from neuroscience drugs specifically.
Okay, that's perfect. Thank you very much for that answer. Now, the next question is wondering, and perhaps Dr. Delat, you can comment on this one. It says, across the various approaches discussed, what are three just non-invasive approaches to manage potential toxicities in clinical settings?
Very good question. When we talk about non-invasive approaches, which, that's the preference, getting the self-report data, where the patient actually gives you insights on what's going on with them. Certainly doing procedures such as MRI, right, where you put them in the machine and you can understand brain chemistry, you can understand what's going on in the tissues. Those are very important and not invasive. The other thing, of course, is limiting the patient population, right, based on your inclusion-exclusion criteria. So that's going to require that we have a very thorough patient history, medical history that's taken. So those are your three examples.
Very good. Thank you very much for that answer. Going back for a moment to Dr. Wakeford, the question here is curious. Does the overall number of patient populations scale with the corresponding drug market for neurological disorders?
Yeah, that's a really good question. And interestingly, often that's not the case. So for example, Alzheimer's disease has around 55 million patients worldwide, and the annual drug revenue for therapeutics that are designated specifically for Alzheimer's indications is around $6.3 billion, at least in 2022. But in comparison, for patients who suffer from multiple sclerosis, there are about 2.8 million patients, and the annual drug revenue for that is about $27 billion. So definitely a gigantic difference in revenue shares there. One of the main differences related to this is partly explained by multiple sclerosis often has an earlier onset, so around 20 to 40 years of age, compared to Alzheimer's, which has usually a later onset around 60 to 70 years of age. But it also it's also due to the fact that there's a substantial gap between the number of marketed drugs that are available for Alzheimer's. So it's about a little bit over 400 brands worldwide in comparison to multiple sclerosis, which is around 189 brands worldwide. So taken together, this sort of implies that the current allocation of drug innovation doesn't really meet or represent the current patient indication needs, and sort of serves a greater purpose as to showing that there is a pretty big underserved CNS drug market currently.
Okay, very good. Thank you very much for that answer. Now, I want to ask a question for Dr. Welty. Someone here is wondering, why were single lumbar punctures used for CSF sampling rather than using a CSF catheter for the clinical PK study? And further, are there any limitations?
Yes, thank you. That's a great question. We originally would have preferred to use a catheter, which would have given us a serial profile from each one of the subjects. However, in our discussions with the FDA, they felt that that was too much of a risk for the healthy volunteer population. And therefore, we agreed on the single lumbar punctures rather than using a catheter. However, catheters are very commonly used and a good tool in understanding both biomarkers and pharmacokinetics in the CSF.
Very good. Thank you. Since I have you, I've got another question for you. Someone wondering what were the differences in PGP efflux?
Yes, so different cell lines overexpress the P-glycoprotein transporter differently. And depending on the cell line that's used, for instance, the Netherlands Cancer Institute cell line has a lower expression than the National Institute of Health cell line. When using these two cell lines, the drug in one case looked like a PGP substrate, in the other case it did not. So it was really important for us to prove in the clinic which one of those cell lines was more predictive for the clinical outcome.
Very good. Thank you so much for that answer. Now, I have another question for Dr. Delat. Someone says that developing a profile for neurotoxicities is a complicated process impacted by various factors. So what they're curious about is what are a couple of examples of how a profile for neurotoxicities observed in a general toxicology study may be leveraged to inform the design of a follow-up EEG study.
Yes. So first example, importance of leveraging clinical signs. So if you observe an animal that's having changes in reflexes, that, for example, may be an indication that there may be a brain lesion, right? And so that would help to inform your follow-up studies, which may use, for example, special stains in the brain to understand if there is any kind of lesion, achy lesions. The other thing is leveraging toxicokinetic and pharmacokinetic data to understand, when the peak effects of the drugs, for example, may occur, to understand when you should do certain assessments. So, for example, you would want to do, for example, video monitoring in or about the time where you see peak plasma levels, right, which could be associated with the exhibition of convulsions. So leveraging TK data can help with the timing of assessments, and leveraging clinical signs may help in terms of understanding what types of assessments may be needed in follow-up studies to characterize your potential neurotoxicities.
Wonderful. Well, thank you very much for that answer, and everyone for all of your answers. However, we have reached the end of the Q&A portion for our webinar today. Now, if we couldn't attend to your questions, know that the team at Allucent may follow up with you, or if you have some further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, there's a link to view the recording of this event, which I've placed in the chat box for everyone. So I encourage you to share that with your colleagues so that they may register for the recording here as well. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you for coming.